introduce you and just let everyone know how lucky we are to have you taking time away from your very busy schedule to join us to speak about the aging brain and HIV's impact on that. And just wanted to hit a couple of the high points from your UCSD profiles page, because I was very interested in your background, actually. I didn't realize all the different groups and departments that you had such an important role with. But basically, it looks like, please correct me if any of this is outdated, but Associate Director of the NIMH-funded HIV Neurobehavioral Research Program, PI of the Neuro Assessment Corps, and also involved as an investigator at the Translational AIDS Research Center and the Center for Medicinal Cannabis Research. And it looks like you're doing a lot of integrated program research on human clinical neuroimaging, lab and animal model investigations at UCSD, and have collaborations with Sanford Burnham Institute and the Scripps Institute, and that you're the chair of the Neurology Collaborative Science Group of the AIDS Clinical Trials Group. So you are a very busy man, and we are very grateful for you taking the time to be with us today. So with that said, I will let you get going. Thanks so much.
Okay. What made you think of me other than my wife, and you couldn't get anybody else to do it?
Well, I think
that's not true.
It's not true at all. And, you know, interestingly, we are collaborating with Rayanne Moore's group, and they are recruiting from our cohort for one of the studies where some of the Alzheimer's patients and the MCI participants are one of the comparison groups in one of her studies on virtual reality. So that's one connection and one opportunity for our team to better expand our awareness of what other groups under the Department of Neurosciences umbrella is doing that has some relationship to our team and our participants.
Yeah, well, I think you'll see a lot of crossover here. And incidentally, we are working with Mark Bondi and Aaron Sunderman, and one of my fellows has a project with the ADRC looking at tau PET imaging. So yeah, lots of overlap here. So I'm going to talk about overlap between HIV and aging, both at the clinical level, biomarker level, some of the underlying molecular biology, and then briefly touch on some potential interventions. Okay. I thought it would be important, given that this group is probably not very familiar with HIV, just to talk very briefly about biology and what happened. I was part of the HIV research arena back when everybody was dying. Basically, nobody lived for very long, and nobody came into an age range where they were at risk for Alzheimer's disease or other neurodegenerative diseases of aging. This is just a timeline of the availability of antiretroviral drugs. There are getting close to 50 of them now. It's a very diverse armamentarium. You'll see in a moment. The idea is to attack at least 2 points in the viral life cycle and to suppress the viral load to undetectable. And we'll talk about the different classes of drugs, and want to just point out that even though we can suppress to undetectable, the virus is integrates into the human genome, and the antiretrovirals can't get at it there, so it's persistent. And basically, as soon as you stop the medications, the virus rebounds to the prior level. So it requires lifelong treatment with all sorts of side effects, as you could imagine. So here are some of the components of the viral life cycle. The virus has to attach to the cell. You're familiar with how coronavirus attaches to the ACE2 receptor. In the case of that virus, well, HIV uses a different couple of proteins, the CD4 and then a co-receptor, either CCR5 or CXCR4. That brings it into the cell where it releases the viral RNA. The RNA then has to be reverse transcribed into DNA. And so the earliest antiretrovirals were the reverse transcriptase inhibitors. And then it has to get integrated into the host genome. Another class of antiretrovirals, very potent and used in most patients nowadays, is the integrase inhibitors. Protease inhibitors basically interfere with the packaging of the viral RNA and proteins to make new viruses. So in addition to all the tremendous benefits with regard to AIDS-related morbidity and mortality, the antiretroviral therapy has been very beneficial for the brain as well. So this is work we do with Ellis or Maslia, looking at synaptophysin on the top and MAP2 microtubule associated protein type 2 on the bottom, and showing that people who die having received suppressive antiretroviral therapy have a better synaptodendritic complexity. But having all these medications and their effectiveness has now converted HIV from a terminal illness to a treatable, manageable chronic disease, in the same way that insulin converted diabetes to a chronic manageable disease. And that has resulted in an increasing number, of course, of aged people with HIV infection. And you can see both in the case of men and women, looking ahead to 2025, which isn't that far away, lots and lots of people in the 55 to 64 and even 65 plus age range. And I regularly see people 65 and over in my clinic. You might be wondering why the difference between men and women. Well, remember that initially the epidemic in the United States was largely in the men who have sex with men population. And then subsequently, it worked its way into women as another vulnerable population, but there was time lag there. So women are unfortunately catching up to men. This introduces the concept of premature aging, and one marker of how HIV is bad for the brain. And the take-home message here, looking at abnormal white matter in the brain, which is one of the more prevalent markers of HIV brain disease. You can see that basically the accumulation starts at an earlier age and it proceeds at a somewhat faster pace, especially during those younger years. So I'm going to talk about lots of reasons why people who are aging with HIV might continue to have neurological problems despite antiretroviral therapy. I recall a lot of people, including HIV experts, saying, 10 to 15 years ago that, well, neuroAIDS is gone, nothing to worry about anymore. It ain't true. And one of the major players is comorbidities. And they have, or helped to explain this gap in mortality and life expectancy between positive and negative individuals. You can see back here is where the potent regimens were introduced, a dramatic reduction in mortality and increase in life expectancy. And that's continued actually to improve. We think because the therapies are more potent, they're less likely to develop resistance, and they're more tolerable, so people take them. In terms of comorbidities, you're all aware that age is the largest risk factor for all sorts of chronic diseases, whether or not you have HIV infection. But I'm going to show you that this effect seems to be amplified or accentuated in the context of HIV. So here's a commonly used sort of cumulative comorbidity burden index, the Charlson. And one of our earliest studies using this scale, we showed that the burden of comorbidities was higher in people with HIV generally. If you looked at this taking all comers, you might say, well, it's modestly increased, but really where the action is in the older HIV positive people. And we're not talking about very old, we're talking about over 50 years. But it doesn't stop there. So if you look at geriatric versus elderly, older HIV positive people, the geriatrics being over age 74, you can see the dramatic increase in all of these comorbidities, or most of them. And if you look particularly at the number of people who have 4 or more comorbid conditions, it basically almost quadrupled in the geriatric versus the elderly populations. So one of the more prevalent comorbidities, both in HIV and in HIV uninfected people, is the metabolic syndrome. People with abdominal obesity, high blood pressure, insulin resistance, hyperlipidemia. And just to give you a sense of how common this is in the context of HIV, we're looking at here at results from a study of over 2,000 HIV positives who were enrolled when they started their first antiretroviral regimens. Already 20% of them had or met criteria for the metabolic syndrome at baseline, and a full 27%, so more than a quarter of them developed incident over just 3 years of follow-up. So very common condition. As you might imagine, age is a strong risk factor, as well as increased body mass index. One of the, I'll be talking a little bit more about this later, but this common manifestation in the context of HIV is increased visceral fat. So this is what we use waist circumference as a marker for this. And the basic idea is that the fat deposition is intra-abdominal as compared to quote unquote normal obesity, where most of the fat is deposited extra-abdominally. And you can see this sort of schematically represented here. This vat is bad stuff. It produces a lot of cytokines that are inflammatory. It is associated with insulin resistance and other metabolic abnormalities. So this is the bad player. Having subcutaneous adipose tissue is not good, but having visceral adipose tissue is really bad. Both overweight and visceral adiposity are associated with lower volumes of a variety of brain structures, especially putamen and caudate, and to some extent the insular cortex as well. All these comorbidities associated with vascular disease are also associated with the increase in neurocognitive impairments prevalence and severity. And it's been looked at a whole bunch of very large studies using a variety of ways of summarizing metabolic syndrome and vascular risk, and they all agree, especially that diabetes is an important risk factor. Here's another example from the same study that I talked about earlier, that study with people enrolling people who are starting their first antiretroviral regimen. You can see that these atherosclerosis and cardiovascular risk factor scores are associated with worse neurocognitive performance 4 years later. So this suggests to me that these are actually causal factors rather than just correlations, although it is possible that there's some third unobserved variable that explains the two together, and it's just that the cardiovascular risk appears earlier than the cognitive impairment. By the way, stop me if there are questions. I think the group is small enough that you can just barge in. So, polypharmacy, as you know, this is a marker of comorbidities. In this study, we did polypharmacy as 5 or more concomitant drugs beyond the antiretroviral. And it's associated with worse global neurocognitive performance in these categories. Interestingly, these other ones aren't particularly surprising, but antimicrobials surprised us. And in retrospect, I think what this may be is just another marker of comorbid conditions, especially infections, and also a marker of having had previously more advanced HIV disease. The domains affected the most were executive functioning and learning. And interestingly, if we account for the comorbidities for which these drugs are prescribed, that doesn't weaken the strength of the correlation between polypharmacy and global performance, suggesting that the drugs independently are contributing to the impairment. These comorbidities are associated with abnormal brain MRI indices, as well as the neurocognitive that I've been showing you. So I mentioned white matter as a prominent focus of HIV-related brain disease. And you can see in this confounded group, marked increase in abnormal white matter. And let me tell you what confounded means. So we do comprehensive case conferences on each research subject, and we come to a consensus about whether any comorbid conditions like a developmental learning disability or head trauma is either incidental or contributed to the cognitive impairment, or likely explains the cognitive impairment. Most of these confounded people have several, not just one, explanatory conditions. And of course, it can be at times difficult to decide, but we look at things like when was the onset of the cognitive difficulties relative to HIV infection. If it's been there all their life, it's unlikely that HIV was the primary contributor. Total white matter volume strongly associated with neurocognitive confounding, but there is signal with regard to both cortical and subcortical gray matter in the people who have only incidental neurocognitive comorbidities. And I want to make a clear distinction between neurocognitive comorbidities and medical comorbidities. So the neurocognitive are the things I mentioned, like head trauma and developmental learning disability, whereas the medical are things like pulmonary disease and diabetes and so forth. We just did this analysis showing that, you remember I talked about this Charlson index, that it increases over time. So here over 20 years of follow-up in some individuals, just a gradual accumulation of these comorbidities. And the people who have more comorbidities have the worst neurocognitive performance, highly statistically significant, and there's an interaction also. So the older you are, or the longer you're studied rather, and the higher the Charlson index, the steeper the decline. So what we think is going on here, I mean, you all have thought a lot about cognitive reserve and education and so forth. And we think that there are a whole bunch of things that contribute to loss of cognitive reserve in HIV. And here's where we see the age-related comorbidities. Probably worth mentioning this sedentary lifestyle. Often patients don't have a lot of choice here. They have painful neuropathy. They have arthritis and lots of things that limit their ability to participate in physical activity. Like other vascular diseases, stroke prevalence is increased in people with HIV. So you see a couple of studies here from back in 2011. And an interesting sort of unique feature, whereas vascular disease in the non-HIV population is much more common in men, as you can see here. The principal difference between HIV positive and negative is here for women. Women have a more than twofold increase in vascular risk compared to men, where it's a much smaller increase and not statistically significant. Here's another way of looking at the same thing. So it's in this 40 to 49-year-old age group where that male-female difference occurs. We think this may have something to do with menopause and changes in hormones right around that time period, because it doesn't persist subsequently. So the kind of things we talk about when we talk about clinical stroke and TIA are related to large vessel disease, either in the neck or intracranially. We do have some ways to look at small vessel disease, but they're harder to study than the large vessel disease, but I'll show you some information about that. So we do have some imaging markers of small vessel disease. So this is a FLAIR image of the brain of a guy who's in his 70s. And you can see here very extensive bilateral white matter disease. This is what I was talking about earlier. But in addition, there are a few focal areas of infarction here, presumably lacunes. We did an autopsy study looking at small vessel disease and specifically amyloid angiopathy. In this condition, you take a normal vessel with lots of smooth, these are middle-sized arterioles and small arterioles. So they have endothelium and smooth muscle, and that gets replaced in milder disease with just partial thickness replacement, and in advanced disease with full thickness replacement. by this hyaline material. This is strongly associated with hypertension and diabetes, but as you can see, it also occurs in HIV. And it occurs in a whole bunch of different brain regions, as you can see listed here. So in our autopsy series, we found it to be either mild or moderate to severe in 3/4 of the brains. In addition to the association with HIV itself, it was also associated with these protease inhibitors that I mentioned earlier. They are a common source of some of the metabolic abnormalities that we just talked about. So they seem to contribute to the increased prevalence of the metabolic syndrome in HIV. This small vessel disease is also associated with neurocognitive impairment with a very high odds ratio. We do have ways at looking at cerebral blood flow that reflects small vessel disease. So this is in people without large vessel arterial disease, showing different ranges of reduction in blood flow in multiple cortical regions. So let me turn to another important area that we think makes a major contribution to brain disease, and that is inflammation and immune activation. So why do these guys, they're suppressed, why do they have inflammation? Well, in part, it's because they're left with a partly deficient immune system. So there is immune reconstitution when you suppress your virus, but it doesn't return to a totally normal cellular immunity. So you get viral reactivation. So more than half of us have CMV co-infection or CMV infection that's chronic. We usually control it quite well, but when you have immunodeficiency, you can reactivate the infection. The patients don't get sick. Just if you look in the blood, they'll have spikes of CMV viremia. And then some of the viral proteins, which despite suppression are still transcribed. So infectious viral particles are not made when they're fully suppressed, but viral proteins are made, and they contribute to inflammation and immune activation. And then there's abnormal microbial translocation. So this is products from the gut microbiota that cross the gut barrier into the circulation, things like lipopolysaccharide. And in the circulation, they are an inflammatory stimulus. So all of these things together increase cytokine secretion causing inflammaging. They increase the risk for stroke and thrombosis that we already saw, and they're associated with malignancies. Here are two very strong and readily available predictors of morbidity and mortality. Interleukin-6 and D-dimer. D-dimer is really a marker of increased coagulation. However, it is strongly related to things like IL-6 and C-reactive protein. I mentioned cytomegalovirus. We control the infection in part by antibody-mediated immunity and in part by cellular immunity. In any case, you can measure concentrations of the antibodies, and among people who are on suppressive antiretroviral therapy, those who have higher anti-CMV levels, immunoglobulin levels, have worse neurocognitive performance. We can measure brain inflammation with PET using these TSPO ligands. These are sometimes done in Parkinson's and Alzheimer's disease as well. And this is a relatively new technology that is a cousin of diffusion tensor imaging called diffusion-based spectral imaging that can be used to image increased cellularity. So there are macrophages and microglia that proliferate in the subcortical white matter and the deep gray matter that can be visualized using this technique. Inflammation is also associated with depression or depressed mood. So this is the Beck Depression Inventory, and we're looking at this is a factor analysis in which 8 markers were included, and one of the factors had these highly inflammatory cytokines, and you can see that when the factor is higher, the mood disorder is worse. Interestingly, this was true in men, but not women, and that has led us to think more about whether treatment of depression, even though it's more common in women, might be focused on inflammation in men and perhaps some other mechanism in women. This combination of inflammation and depression is particularly bad together. So if you've got a high BDI in the red here and your plasma C-reactive protein level is higher, you get worse cognitive function. So some of you may be aware that a guy here in San Diego named Gerald Chun recently published in Science this mechanism that might explain amyloid deposits in people with Alzheimer's disease without HIV infection, and the possibility that using these reverse transcriptase inhibitors could protect against that. I'm going to argue that this is not a significant issue, but let me explain what we think is or what Gerald thinks is going on. So the idea is that the APP gene is transcribed into mRNA and spliced, and then there are reverse transcriptases that are part of the genome. They're basically old retroviruses that got permanently added to the human genome, and there's actually quite a lot of DNA that is these old retroviruses, and they do describe reverse transcriptases. So the mRNA gets transcribed into reverse transcribed into a DNA that then reinserts itself back in the genome. And now reverse transcriptases are very, very sloppy copiers. So they introduce things like single nucleotide variants, insertions, deletions, and so forth. And when those go back into the human genome and are transcribed, they produce amyloid that contains those same mutations now, and that can produce APPs that are deficient, and in some cases that have the same characteristics of the APPs in familial Alzheimer's disease. So that's the concept that why even somebody who doesn't have one of those mutations could actually sort of acquire the mutation and then produce the abnormal protein. But here's a case. This is a patient of mine, 73-year-old, high functioning, 18 years of education. You can see that, as I mentioned before, he's got all those risk factors for vascular disease, high blood pressure, hyperlipidemia, diabetes, and so forth. He doesn't smoke, and he's got a real, very supportive family, fortunately. On the usual medications, in addition to his antiretrovirals, and I want to take particular note to point out that tenofovir and emtricitabine are reverse transcriptase inhibitors, and virtually every single person with HIV is on a reverse transcriptase inhibitor. They're just part of the standard regimen. So what Gerald is hypothesizing is that, well, those same reverse transcriptase inhibitors are going to inhibit the reverse transcriptases that come from the endogenous retroviruses. So I first saw him in 2016, and he was describing something that sounded more not like progressive cognitive decline, but instead transient lapses of attention. You know, we considered the possibility that these were seizures, that he had Lewy body disease, or that they were related to cerebrovascular disease. His mini-mental was fine, as you might expect for a guy with high education, high functioning. He was suppressed and had a very nice CD4 count and antiretroviral therapy. And so we did a workup, had him continue his aspirin, wanted to get good neuropsych testing, and did some vascular imaging, and wanted to check his spinal fluid viral load. So the standard is to check the blood plasma viral load, but it's also possible to measure the virus in the spinal fluid. And I won't go into this in detail, but there are case reports and small series of patients who have undetectable plasma viral loads, but still have detectable CSF viral loads, and they can be symptomatic. We subsequently showed that, in fact, the vast majority of them are asymptomatic. And the way we do that is we just do lumbar punctures in the context of research studies in otherwise asymptomatic people. So here's his brain MRI, very similar to what I showed you earlier with the extensive white matter disease, lacunar infarction here. As a matter of fact, this is the guy I showed you previously. It's just a slightly different level. So we concluded on the basis of this, his carotid duplex was unremarkable, that this was likely to be cerebral small vessel disease. And he went on to progress, however, so converted from this transient lapses of attention now to trouble recalling names, had a couple of accidents while driving. His mini-mental state score declined a little bit, but he didn't yet meet criteria for dementia. When he finally had the full neuropsych testing a month or two later, he clearly was moderately globally impaired, multiple domains, evidence of his high premorbid ability in the wide range achievement test score. And his worst deficits included delayed recall. So a little bit of a clue there that some kind of Alzheimer pathology might be playing a role. He had relatively intact working memory and motor functioning. So unfortunately, he continued to progress. Here, they said he was still driving and handling his finances, but now his MoCA was clearly lower than that mini-mental state that I showed you before. And we felt at this point that there was enough evidence to consider that Alzheimer might be contributing to his cognitive progressive now cognitive decline. So we gave him some donepezil. Then the family said he's getting much worse. They wanted to take away his car keys, but he was very, very reluctant to do so. His MoCA declined even further. His neuropsych testing showed moderate to severe rather than moderate. And we held a family conference, and I told them that I thought this was Alzheimer's disease with a possible additional contribution of cerebral small vessel disease. We did a lumbar puncture, and he fell right into the quadrant consistent with a diagnosis of Alzheimer's disease. We were unable to get his insurance to pay for a PET scan, amyloid PET, but unless somebody else wants to argue with me, I think this is pretty solid evidence that he does indeed have Alzheimer's disease. Okay, so is there any evidence of abnormal amyloid processing that might contribute to an increased risk of Alzheimer's disease and HIV? The answer is yes. So you may be aware of this TREM2 triggering receptor expressed on monocytes, I believe, type 2. This is something that helps microglia clear A beta from cells. And it turns out that it's reduced in the brains of people who've had HIV-associated neurocognitive disorders. And it's thought that the activation of those microglia interferes with the function of microglia in clearing A beta. And some of you may recognize this. There's a TREM2 variant that has is a very strong risk factor for Alzheimer's disease in uninfected people, similar actually to ApoE4. We've looked for amyloid in the brains of people with HIV, compared it to a general autopsy cohort. In fact, there is an age-related stair-step increase in the prevalence of amyloid plaques in people with HIV, being higher than in people without HIV. We've done spinal fluid studies where we were able to show that levels of A beta 42, as you know, decreased in Alzheimer's disease, are intermediate between AD and controls. And you can see also that there is no elevation in tau levels, which is curious. In fact, they're somewhat decreased. Here's another mechanism by which cognitive problems might happen in HIV. So you may be familiar with these protein quality control pathways. The PERC is one that when abnormally folded proteins start to build up within the cell, this system turns on and it blocks translation of new proteins. So it's a kind of a beneficial response to the formation of the abnormally folded proteins. However, if that remains turned on, it's obviously going to be damaging to the cell because it won't be able to make the proteins that it needs. And it turns out that there are some variant single nucleotide polymorphism variants that confer an increased risk of progressive supranuclear palsy that also seem to be associated with cognitive impairment in people with HIV. So can we do anything about all these bad stuff going on? I'm not going to talk about all of these things. I'll mention tesamorelin and then these senolytics. So tesamorelin has to do with this issue that I described earlier, the accumulation of visceral rather than subcutaneous fat in people with HIV. So the visceral fat is a bad player. It increases insulin resistance. It produces cytokines, inflammatory. And tesamorelin is an analog of human growth hormone releasing hormone produced by the thalamus and stimulates, of course, growth hormone, which is deficient in people with HIV. Tesamorelin selectively reduces that visceral rather than the subcutaneous adipose tissue. And we believe it will improve cardiometabolic mediators like hyperlipidemia that might favorably affect brain function. But a second, perhaps as or more important mechanism is that it stimulates the liver to synthesize and secrete insulin-like growth factor type 1. IGF-1, there are receptors on brain endothelial cells that actually recruit IGF-1 into the brain tissue, and that's how important it is for protection of neurons. Among the things that it does is stimulate brain blood flow, protect against oxidative stress, improve glucose utilization, and overall higher levels of this IGF-1 correlate with better cognitive performance. You may be interested to know that this same drug was studied in the context of older people with about half of whom had mild cognitive impairment and were at risk to progress to Alzheimer's disease. There was evidence of a positive signal. It was only a phase 2 safety and preliminary efficacy trial, and the people who made the drug didn't elect to pursue it further. But as I said, it's already approved for this visceral adiposity in HIV. So we simply hijacked that to do a study of cognitive function. And that study is ongoing. We're having trouble recruiting, but we're about 60% of our full accrual. So what about these senolytics? What does this have to do with? Well, as people age, senescent cells accumulate. So these are cells, including immune cells and cells in other tissues, that are basically should have died, apoptosed, but they get arrested in that sequence and they just hang around. They don't do anything useful. And in fact, they do produce toxic things, chemokines and cytokines and so forth. And so there are these drugs that can basically propel them on their way to death. And those are called senolytics. They lyse senescent cells. And these senescent cells are markedly increased in prevalence in HIV. So we think that they may well contribute to brain disease in HIV. And there's no trials ongoing yet of these drugs, but there's a lot of interest and enthusiasm in them. And that's basically what I have. Just some pictures of collaborators, some of whom you might recognize here. Let's see, David Moore, Eliezer Masley, of course. Let's see who else here. Aaron does some work with the ADRC, I think. And Adam is working with Chris Akeem in the experimental neuropathology lab. Soon did those cerebral amyloid studies, amyloid angiopathy studies that I talked about earlier. So, questions? We got plenty of time.
I did see one in the chat. Someone asked if the prevalence of HIV and neurological disorders manifests differently in different ethnicities.
Oh, so one of these people, Maria Marquine, is very interested in that topic. And basically, she's got grants to look at. The answer is yes, but she's interested in knowing whether health disparities, so less effective healthcare in Hispanics, might account for their increased risk of neurocognitive impairment.
That's great. Please feel free to just unmute yourself or, oh, okay, I see another one submitted. Can I draw a conclusion to say HIV does affect AD progression? Was one of the questions in the chat.
I don't know about AD progression. I'm convinced that despite what Gerald Chun has proposed, that AD does occur in people, and it occurs in HIV patients who are typically on these reverse transcriptase inhibitors and have been for decades. So these are just part of standard therapy.
Ron, Howard Feldman here. Can you hear me okay?
Yes. Hey, Howard.
Good. Thank you for this nice talk. So one of the conundra that I see with this is a strong relationship to amyloidopathy and a weak relationship to tauopathy.
Yeah.
And tau is, of course, and tau pathology is the signature and, in fact, probably a critical piece in the Alzheimer pathogenesis. So what I'm wondering is this an amyloid inflammatory effect that does not run through tau, and hence we will have trouble identifying it as typical Alzheimer's disease?
I think that's entirely possible. Yeah. I mean, I showed you the data. It seems to be amyloid more than tau that is affected. Some people have found tau in the brains, but the evidence is less strong.
Because it sounds like you can get up to intermediate levels of pathology, but BRAC 5 and 6 less so.
Mm-hmm.
That there's, I think, the TREM and the microglial activation and probably astrogliosis is a considerable effect and perhaps part of a mechanism in play that could be cognitively difficult for the brain to contend with.
Exactly. So, you know, you've got an already vulnerable brain with all these comorbidities. It wouldn't terribly surprise me if the course of disease was accelerated when people do get Alzheimer's. And there are other mechanisms that might increase amyloid production. So one of the HIV proteins, the transactivator of transcription protein, is an inhibitor of neprilysin, which, as you know, is one of the amyloid cleaving enzymes. So that's another reason why amyloid might build up.
So Tesmorelin sounds very interesting. How do you gauge target engagement and the sufficiency of the dose level?
Yeah, so we are using the dose that's FDA approved. That was sort of we couldn't do much about that. But the dose we're using is higher than the dose that was used in that prior MCI no HIV study.
Mm-hmm.
And it's given for longer. And we are measuring several markers of its effectiveness, of course, the visceral adipose tissue itself, but we're also measuring IGF levels. And we're also measuring hippocampal volume and measures of MR spectroscopy that are associated with inflammation in the white matter and the basal ganglia. So I guess the best target biomarker is the IGF-1 levels.
Right. I'm wondering if ADCS should be looking at trials with tesamorelin.
I don't know why. You probably know Suzanne Kraft. She
Yes.
was one of the people involved in that non-HIV study. I just don't know why she didn't pursue it further, because it did seem to work, and it's a pretty darn well-tolerated drug given to thousands of people or tens of thousands of people. So,
Cool. Okay, something to chew on. Thank you.
you bet.
There are any other questions? I think we're going to wrap it up soon, so please feel free to hop in or type it into the chat, and I'm happy to read it for you.
Too bad Sarah Banks isn't there. She's working with Monica Diaz, who's now in Peru, looking at tau PET. We're trying to get that going in some HIV and AD patients to compare, look at that question that Howard raised of the tau levels.
Great. Well, thank you so very much for your time and for putting together such an excellent talk. We really appreciate it. And if there are
My
no
pleasure.
Thank you, Ron.
You bet. Take care all.
Thank you so much.